Right. It's Friday. 3 weeks down, 12 weeks left. Who's counting? Tell you what, as soon as the GS 1181 course is done, it feels like it's all done. It's like I feel like I've got loads of time. But anyways, all right. So reminders, quiz. Check it out. Get it done. We'll have a quiz on rounding and these frequency distributions coming up. That'll go live probably on Monday.
How far does the quiz go based on my PowerPoint?
Rounding plus. Oh, the quiz went up to rounding. There's no rounding. There's no rounding, and the majority of it is like variable types and variable scales. Variable types and variable scales. All right, and then we'll have homework. You've submitted it. I've read through it. We did have a couple people that did not submit the homework. All right, so had the due dates. I didn't look super closely at it. I kind of scanned through, looked for, make sure you had the correct figures, make sure you were getting some output, correct output. Again, it's graded. It's did you do it? Did you not do it? Or is it halfway complete? I will get the key posted, so then you can look at how I did it and compare it to how you do it. All right. The only difference is there will be a couple places where there are multiple ways to accomplish the same task, and usually I address that in the homework and say, okay, here's one way to do it, here's another way to do it, either way will work. All right, so we'll get those posted. All right, so we left off with the rounding. We did this, right? Went over the answers for all of these numbers in the implied range. Is that right? So, all right. Talk about derived variables. All right, so these derived variables are computed from two or more independently measured variables whose relations are expressed in a certain way. All right, that whose relations are expressed in a certain way is basically however we are taking this derived variable. So surface area to volume ratios. Surface area is measured independently, volume is measured independently. Taking the ratio is that expressed in a certain way. It's expressed as a ratio. All right, you can say area. A surface area is a derived variable. All right, it's a length and width type of variable. If it's like rectangular, that's your quadrilateral, right? It's a quadrilateral, it's length and width, right? So if we have independent measures and then we do some sort of calculation with them, multiplication, addition, subtraction, multiple, did I say multiplication? Multiplication, division, addition, subtraction, then we're working with the derived variable. All right, benefit of this is that we're taking two or more of these variables and condensing them into a single variable. And we're hoping that single variable captures what we're trying to discuss. So surface area to volume ratios are about heat loss or water loss. All right, that's a little bit better. It tells us more information about those biological processes than just surface area or volume alone. All right, if we're looking at overall size of an individual, we could take the height, or we could do the weight, or we could do this composite weight, basal body mass index, BMI, all right, to get our overall healthy size. And the reason I say weight versus BMI is because if you're taller, you're going to weigh more than somebody that's short. So if you compare weights and say, well, the more you weigh, the unhealthier you are, Well, that kind of goes out the window if you're comparing somebody that's 5 feet versus someone that's 6.5 feet. Right? That relationship goes out, but this derived variable, body mass index, you might say it's not the best, but it is more predictive of overall weight than just weight itself, or healthy size than just weight itself. Light capturability is the other thing. We can do length, we can do width, or we can do overall surface area. Surface area is a little bit better because you can have a really long leaf, but just be really thin, say like a needle. That's going to have a low capturability. You could have a leaf that's pretty short but fairly wide, say the ginkgo leaves. Right? Surface area is going to be that better estimate the light capturability. So these are derived variables. All right. Some of the common types of derived variables would include ratio, index, and rates. Ratio, don't confuse this with our ratio scale. All right. With the ratio, we're just taking one value, dividing it by another, or doing some sort of numerical, or let's say addition, multiplication in the numerator, and then addition, multiplication stuff in the denominator. We're basically getting a ratio. And that's going to show one value expressed, or it's going to express one value in relation to another. The most common ones that we do, surface area to volume ratio, we mentioned that. Sex ratios, all right, is another one. Or we'll hear about variance to mean ratio. The variance of a value of some measure compared to the mean of some measure gives us some indication of spatial patterns and temporal patterns. We'll work with those. So usually when we do ratios, we scale it right to 1 and then express it as a single number. So like with this example, we have 59 females and 48 males, right? We could say, well, we've got 59 females and 48 males, but if we are trying to do a ratio, how would we reference it? Use our calculator. 59 divided by 48. So ratio is 1.23. All right. Now you could say, well, 1.23 what? It's basically telling us, the ratio is telling us we have 1.23 females for every 1 male in our population. All right. If they're the same, it's 1 female to 1 male. We have fewer females relative to males, we're going to be less than. And then we could also report it as a percentage. So instead of doing this 1.23, we could do percentages. So we can say we have 59 females divided by, is that 107? Yeah. We got 55%. So a percentage like this would also be considered a ratio. It's also derived here. An index usually have index in their name, but index could generically refer to derived variables. So like a ratio could be an index, sex ratio index, right? It's just a generic term. But you will see some people restrict their usage to these things. So standardizing of a variable is oftentimes an index. If you combine variables into like a weighted average, or if you include it in some other function, that could be considered an index. Or if you sum up some numerically scored properties, that's also an index. So that Likert scale, the satisfaction scale from 1 to 5, one way you could do it is pick out the questions that pertain to, let's say, taste, all right, overall taste. And you take those questions and whatever the scores are, you just add them all up to get a single index, which would refer to satisfaction with the taste. And you would do the same thing with, let's say, service. And then rates would be our last one. The difference between index and ratio is that the rates always have some value per some unit time or per unit area or per unit volume. All right, so speed, that's a rate because it's measuring your distance traveled per some item, miles per hour, miles per minute, however you do it. Total time to complete a race, total time to complete a task is also a rate because it's per task or per race and so forth. All right, does this matter? Am I going to ask you, is this a ratio, index, or rate? No, I'm not going to ask you any of this. I'm presenting these for completeness because the reality is you're going to analyze these things especially index and rate, in exactly the same way. You take that value, that's the value that you use in your t-test and your ANOVAs and so forth. Ratio is a little bit different because with ratios, we probably violated an assumption with the ratio, especially if we work with percentages. So those are kind of like the exception, and I don't know if we get to it or not, but we'll talk about it briefly. All right, so what's the advantage? We already said that. Oftentimes it's the best way to interpret or describe some biological process. All right. Disadvantage, though, of these is that they tend to be inaccurate. All right, because what we have to do is take into account the implied range for each of the independent variables that are included in our equation, in that function. All right, and when you do that, anytime you do that, your implied range of that derived variable is going to, not the implied range, the error part of that range is going to magnify and increase. All right? So here's an example of surface area to volume ratios. All right? We do a surface area of 3, volume of 5. We do the ratio, and we get 0.6. All right? So that 0.6 implies that the actual surface area could be anywhere between 0.55 and 0.64, right? That's our implied range. That's actually wrong, because our implied range refers to those variables that we measured, which would have been surface area and volume. All right, what we need to do is recognize that the surface area, this number 3, could be anything between 12.5 and 3.4. The 5 could be anywhere between 4.5 and 5.4. So then what we have to do is take, our smallest number here divided by the biggest number here. That's kind of represents the smallest value that we can get. And our upper end is the largest value that we can get, which would be this 3.4 over the 4.5. So our actual error range for our surface area to volume ratio would be 0.46 to 0.76. Notice how much larger it became. All right. This is a width of like 10 units, or 0.1 unit. All right. This is like 0.3. Is that right? Yes, 0.3. That's pretty big, right? So that's when we say it's relatively inaccurate. Same thing if we did surface area. So actually, this is a little bit incorrect because we calculate surface area using lengths and widths, or if they're circular, you can use some elliptical equations and so forth. So yeah, let's say this was 3 by 5, okay? 3 length, 5 width. You have the same sort of situation. If you took 15, you would assume this is the implied range. So our surface area would be between these two values, but they're actually wrong. We have to take the implied range of each of these, and this one gives us now a surface area range of 11.25 to 18.36. So there's a lot of things we have to consider with the derived variables, including the possibility that our actual error rates are going to be much, much higher. Second disadvantage is that usually these derived variables don't follow a normal distribution. And if they don't follow a normal distribution, then we really can't use the classical tests that assume we follow a normal distribution. All right, normal distribution, that's our bell curve. All right, that's going to be one of our major assumptions for all of our tests, because if we follow a normal distribution, then some of our equations to get our test statistics will match the theoretical distribution of those test statistics. We'll talk about assumptions and why they're important. Second, they don't really reveal anything about the relationship between two variables. All right, we work with the surface area to volume ratio. It doesn't really tell us if surface area increases and volume also increases. It doesn't really tell us that correlation. So oftentimes, if we're doing surface area to volume ratios, we look at both surface area and volume to see what is that relationship and get a better idea about it. And then also, lastly, our frequency data or counts might actually generate a continuous distribution. So if we did, I'm going to use dice, right? Who has at least heard of craps? Casino game? Actually, you all have probably played Monopoly, right? Right? With Monopoly, you roll 2 die, figure out how far you can move. Can you roll a 1? No. Individual die, you can. But when you take the derived variable, which would be the sum of those, all right, you can't roll a 1 anymore. Just impossible. All right? And that fact alone might actually violate or might cause us not to get this continuous distribution, along with possibility of getting multiple values in the same combination. So there's 6 ways to roll a 7 in Monopoly. 7 is the most common number that you'll roll. All right? Again, dice example. So when you have these frequency data and counts and you start to utilize them in some of the derived variables, oftentimes we're going to have to account for this possibility that we're not a continuous generation in our analysis. And I put here solution to transform data to inverse normal probabilities. Don't have to know that at all. All right. All right. Questions on derived variables? Yeah.
I just have a question, like, how are you supposed to know the implied range? So, like, if they give you a different number.
Sorry.
So, like, if they gave you, like, 6 over 12, how do you know, like, the range of 6 and the range of 12?
So the 6, you assume that is their rounded number. So then you have to think, what numbers could round give me 6? So for 6, it would be 5.5 up to 6.4. So if we round to get to that number 6, those are the numbers that we have.
Okay, that makes sense.
All right? All right. So now we're going to get to the first processes of our data analysis. All right, this is kind of leading in. This will lead into a lot of our descriptive statistics and then ultimately our inferential statistics. So here we have some research project. We collect our data, and then you look at it and say, okay, now what do I do? Or your professor says, here's some data. You're going to look at it and say, okay, what's next? One of the first things that we do when we receive these data is to go through and just visualize it. Make a graph. Make a table, just to see what do the data look like. And normally what I like to do is to do one display per variable, just to check what's the ranges, what's the medians. And in a lot of cases, since these are data that I input into Excel, I want to check to make sure that I type the numbers correctly. Right? Because if I'm measuring snails, our snail lengths are going to be between maybe 4 millimeters and 13, 14 millimeters. In length. So I'm using that decimal place. If I do a single graph for that size variable and I see this point way out here, I probably screwed up and forgot a decimal point. So I can go back and look at it real quick. All right, so when I'm doing these variables, we're looking at a couple things. 1, are the data normally distributed? Does it look like a normal curve? 2, are the data skewed? Are we slanted towards one side or the other? Do we see 2 or more peaks in our data and so forth? We're looking for these things along with the possibility that we screwed up typing data in. I had a grad student, that's exactly what happened. We started looking at some of this data. He typed in 1 extra 0. We had a fish that was like massive in his data set. I'm like, I doubt you caught a fish that big. Well, it's in there. Go back and check your actual data book. And yeah, sure enough, extra number, that happens. If he would have done this, he would have caught that. All right, so collectively, when we do these 1 displays per variable, these are called frequency distributions. We're just looking at frequency. That's it, just to check these types of questions. All right, so a frequency distribution is basically taking our data, arranging them from smallest to largest, and putting in the counts. How many are in each category? So for qualitative data, maybe you can't put in order. That's fine. What you're doing is taking every group that we have and counting up how many are in this group, how many are in this group, and so forth. If we're dealing with numeric data, then what we do is usually group them and say, okay, from 1 to 3, this is how many I have. From 3 to 5, this is how many I have, and so forth. All right. Our frequencies, we can report it as either a raw count or a percentage of the total. It doesn't really matter. We're going to get the same looking curve or same looking graph or even a similar same looking table from this stuff. So when we take these numbers, these frequencies, if we make them as a table, we call that table a frequency table or an enumeration table. I think I tend to use frequency table just throughout the class, but know that enumeration table is the exact same thing. If we report these numbers in a figure, then it's going to be one of two types. It's either going to be a bar graph or it's going to be a histogram. The difference between these two is based on the type of data that we're looking at. If it's qualitative data, so if it is categories, we're using a bar graph. If we're working with numeric quantitative data, continuous or discrete data, then we're going to make a histogram. All right? So what we're going to do is we're going to learn how to make some tables and make some of these basic figures. So we have two different data files that are on Blackboard that you're going to download. One is called Freq Table Data, and the other one is Quant Freq Table Data. Frequency. All right? So go ahead and download them, and you can have R Markdown open. I think this one, I didn't say this. This was data in file. There. Let's get my R Markdown. So I have them on these two slides. I have you reading these data in as either qual data or quant data. Import these. Did you get it? So I am going to put up my R Markdown. So we're going to be using these two data sets. We'll probably continue this on Monday. So I just went ahead and as I finished up, this is where we had the implied ranges and the rounding. So at the bottom, I've got visualizing frequencies. I made a header which was reading in data. And when I imported my data, the import data set, what I did was copied and pasted them both in this chunk. So when we start on Monday, all I have to do is go run this chunk, and it should work. Knock on some wood, because who knows what's going to happen. IT might decide to upgrade these computers, and then everything goes to crap. All right. So I'm going to say tables or bar graphs are the preferred method for qualitative data. Tables work. It kind of depends on how many data, how many different categories you have. Oftentimes, I combine my qualitative data with additional descriptors. So, an example for us would be number of fish that are infected with various parasite species. So my table, my frequency table would have the number of fish for each of our parasite species. That's my frequency table, but then I add on to it to say, okay, this parasite is in, this is the classification. Maybe it's a digenetic trematode, maybe it's a nematode. I might have a column that says where that parasite is found, whether it's an adult or larval stage, maybe give a citation to the life cycle, who knows. But the start of the table is our frequency table. Could I use a bar graph for that? Yes, you can, but that bar graph is limited to just the frequencies itself. All right, so let's move this over. All right, so the way to make this table, and to make a pretty table, this is going to be my suggestion, is to get the data into R, have R do the summary for you, and then transfer those data into Excel. All right? Because R isn't really going to make a nicely formatted table. So up here, you can see this is actually a pretty nice table, right? I'd say this is something that you could easily put on a poster or include in a presentation, no problem. That looks good. What R will give you is something like this, right down here. So this isn't something that you would copy and paste and have in your presentation, or at least you shouldn't, because if I see it, I'm going to look at it and say, well, you took the easy way out. Right? You must have been pushed for time trying to get this thing finished last night. So how do we do it? How do we make one of these frequency tables? Well, the function's pretty easy. Table. All right. Now our function needs a vector. All right. Tables. So our function for producing one of these frequency tables is table. And what we have to give it is a vector. So if I look in our qual data set, I can see that our one variable is called make. Make is our vector. That's what we need. So how do we get a table from that vector? Well, whoops. Hold on. It is my shortcut. Control Alt I. Didn't I want to do it? I did. Maybe I got something else. That shortcut wasn't working for me. Maybe I wasn't clicked on. All right. So table, our vector is make, but again, we have to tell R where to find that. That is actually in qual data. So we tell R, give me a table, which is a frequency table, of the make variable. The make variable is found in qual data, and when I run it, this is what I get. This is great when working in R. This is a great check because I can look at it and I can see if I have Chevy, Ford, GMC, other, Toyota, and then I have like another Toyota with like count of 1. You're going to look at this and say, what's going on here? Well, chances are when you input the data in Excel, that 1 count had a space before the Toyota. All right? Because again, the computer doesn't remove those leading spaces. Those leading spaces tend to be counted. So, or if you use different capitalization, you'll see this. This is all something that you would get in R. Now what do? We have these data. Our next step, take it over to Excel to make our nice figure. You can say, well, can't I do it in Word? I'd say, yeah, you can. It's harder. It's a lot harder if you ask me. And at least a lot of the journals, when you submit your papers, they want the tables as a separate file. Or they can ask tables for a separate file. So I'm going to go through here. Let me zoom in a little bit. All right. R went ahead and gave me these in alphabetical order. All right, so what I'm going to do is get this into a table that looks good. So I am going to put these in alphabetical order with the exception of other. All right, I don't want other between my makes. We have named makes. We've got Chevy, Ford, GMC, Toyota. I want the other to be at the end. All right, then I'm going to enter this in. All right, but I'm not done. I need to make it look pretty. Shortcuts, they don't work. All right, so the headers, I'm going to make them bold. I'm going to center all of our values. All right, I want a total column. Okay, so I can add these up, or I could use the function sum. Summarize and then highlight the parts that I want. Or I can pull out my calculator and just do it by hand, no problem. All right, make sure that gets centered. And then I'm going to add my lines. I want a top and bottom. So generally, you have top and bottom order. We want a border at the very top of the table. We want a border at the very bottom of the table. And then you can use a horizontal line to separate the header from the body. And then also to separate that total column. So I'll add these, top and bottom. And now we have a table that looks a whole lot better. A whole lot better. All right, I flew through this. I know that. I'm not asking you to make pretty tables, but I'm giving it to you that you know how I would do it. All right. So there are some rules, and each journal is going to be a little bit different. So what I'm doing is making these tables and structuring them how the journals that I'm most likely to send papers to, how they accept it. There are some medical journals where they want it in a slightly different format. They actually want, shading in the rows and everything. But here's some advice when you make these tables. One, put your categories in a logical order. Alphabetical is great. All right. Maybe you're going to group it by who's doing some independent research. What are you doing? Okay, Gene, differentiate between two species of rabbit. So you're probably using different primers. Okay, so maybe you group your primers by size. This will produce, I'm getting, these primers should amplify a 300 base pair section. These should amplify 400 and so forth. So maybe your logical order is to do it based on size of the fragments that you're screening. And you report then, whether it worked. All right, so that's just general table construction advice. If you have other, you typically put it at the bottom, right, instead of having it in the middle. With your tables, you put your lines at the very top and lines at the bottom. That kind of marks the tables, the beginning and the end of the tables. You don't put lines between every single row. Just excess ink, and it tends to make things a little bit harder to read. You do, though, use a line to separate header from the body. Or if you have a total column, separate the total from the body. And then your total row is optional. All right, you can use a line to separate it, or we can do like alignment to separate it from the rest of the table. That can kind of show up if you have everything lined up here, and I could do, move this over, I can do like a right align for my total, and maybe I could even do this, total equals, so I'm aligned right, and then make that aligned left. So something, it could look something like that. All right, these are just general rules. I put it here so because you may encounter it. All right, but key point: table or generator of frequency table. Sometimes you're going to collect data on discrete data or discrete variables. So this would be quantitative variable. Sometimes you're going to record it as a frequency table. All right, you're going to do the same process as you did for a qualitative variable. So what types of data are these? Usually they're counts. All right, this was, I don't know if I made up these data or I pulled them from another lab, but one of our ecology labs looks at spatial distribution of ragweed. All right, so we dropped grids, we counted the number of ragweed in each grid. So some grids had 0. Actually, in this case, 412 grids had 0. One grid, or 163 had 1 plant, 104 had 2 plants, and so forth. Those are frequencies. We have them in data as each grid represented a row, and then you had the number of ragweed found in that grid. So we had, I don't know how many we had, 700 or so. But anyways, same process. You get your data into R, you make your frequency table, you let R do it. R is really good at doing these counts, making them right here, like this, but R doesn't do a very good job at making them look pretty. All right, so go ahead, get your table, do this as an example, and make sure you get those same type of data. Or the same counts. Oh, because you didn't change the name of qual data or quant data. You read this in as qual frequency table data. Yeah, because, well, look at it. So now we've got our categorization. But yeah, I was looking for make in that data frame, but we don't have that data frame. So if you were going to use it, you would use whatever that name is, qual frequency table data, $. That would work. But in my presentation, I read it in as qual data and quant data, all lowercase. So if you change it in your read CSV chunk, then just rerun the chunk, and you'll see these these data frames appear in the environment. Let me get my R. So if you change this chunk to say qual data and quant data, all you have to do is reread the chunk, and you'll get these to appear in the environment. Yep. So we don't need to make these tables in Excel. Are you taking it for this class for honors credit? Are you an honors student?
Me?
Yeah.
No.
You guys are, right? You taking it for honors? Who's taking this class for honors?
Wait, I'm not taking it for honors, but they don't want honors credit during first semester.
So you're taking it? You're taking it
I'm taking it for credit.
for honors credit?
Yeah.
Okay.
I think she's not.
So I mean, there's a possibility. So I don't remember how many signatures. But there's a possibility that you might present a frequency table where you'll need to make it in Excel. Yeah. So qualitative data. So I'm going to add here tables, quantitative data works best with discrete data. The example that we have is quant creep table data. All right, so I read it in as quant data. Plants is the vector. All right, now I'm going to say something here and add it because in my notes I said works best with discrete data. This can work with continuous data as well. All right, definitely can work with continuous data. Problem though is that continuous data usually has many more numbers associated with that. So like in this data set, the highest that we saw was 9 plants in a grid. That was the largest value. In a continuous variable, if we go to the 10th digit for 1 decimal place, you can have anywhere from 0 all the way up to 9 and every 10th in between. So you can get this massive table just because you're counting up all these fine details. Just as a example, I would put this in here. All right, I'm going to go to this console. Here. Let me make this randomized vector. We'll learn how to make some random variables, but here's an example of a continuous variable with a table. Look at how many we have because it's giving us a count. We rounded, so it's giving us a count of each item. And you see we have a bunch of items that only appear once. That's a consequence of a continuous variable. So this type of frequency table, if we have quantitative data, is best used for discrete data, the discrete data type. So avoid using frequency tables with continuous data. Instead, use a histogram for this data type. All right, so just kind of getting that into our notes so that we have it. So if you have a question, okay, and I say, here's our data. This is continuous variable, and I say, what type of display is best for this variable? If you go to your notes, you'll see this. Oh, okay, so I have continuous data. Frequency tables aren't preferred. So what's best would be a histogram. That's our answer. And you'll have questions like that. What display should we use? What's the best display? All right, questions? Bar graphs. Sometimes we want to make an actual figure, right, instead of a table. Can R do this? Yes. Can R do it? Yeah, R can do it. And we do have freedom in R to modify it. All right. You're probably more used to doing this in Excel. So if you're making a publication quality bar graph, I'd probably just make it in Excel because you've probably done it before. All right. Bar graphs you only use with qualitative data. I know I violate that in the ecology class, but in the stats class we can't. All right. And even in a publication you can't. I would call you out on it. Say that bar graph isn't appropriate for continuous data. All right. The reason why is the information in these bar graphs is conveyed by the height of the bars, the area of the bars. All right. We need that zero. We need that zero to convey that information. All right. So if we're looking at mean lengths or mean heights, you have to start at zero. And I see a lot of people change it to where they condense the graph where the range is only on, let's say, 36 to 45 because your average is 41. All right, that's not appropriate. That's not how the information is conveyed. So only use these bar graphs with qualitative data. All right. The spacing between the bars implies that categorical nature. And our height will refer to either the percentage of the total or the actual raw counts, how many we obtained. How do we get it? Well, the bar plot function. But the bar plot needs a table. It needs a frequency table itself. So in order to make these bar graphs, what we would have to use is first calculate our table and then pass that information to our figure. So figures, qualitative data. If a figure of counts is desired, then we make a bar graph. Oops. See that function is bar plot, table, and then vector. So we generate a frequency. So what this is doing is we generate a frequency table for our vector, and these numbers serve, and actually this table serves as the categories and counts used by the bar plot function. All right, so our example here is bar plot. Bar plot, table, qual data, $fake. And when I run that, I get my bar plot. All right, so just make sure you run this and you can generate a bar plot. And what we'll do on Monday is talk a little bit more detail about how to fix this issue of having other right in the middle of our figure. How do we go about doing it? How do we fix that? Scroll up. There we go. Yeah, because reordering is something we'll do all throughout the class. We'll learn it, we'll practice it a bit. That might appear on like the exam. All right. So if you can make this figure, make sure you save your RMD file and have a good weekend.
Where are you?